Chibo is a researcher at UNAM Sam Nujoma campus in Henties Bay, and she has also previously worked at Ministry of Fisheries. She completed her PhD in Germany, working with institutes such as the Max Planck Institute, the ZMT, that's Centrum for Marine Tropical Ecology, and also the IOW, the Baltic Sea Research Institute. She has a lot of experience in nutrients and in phytoplankton, as well as carbonate chemistry. In fact, she was a previous co-chair, Ocean Acidification Africa Group, which was the African branch of the Global Ocean Acidification Observation Network. So with that, I look forward to Chibo's talk, and I hand over to her.
Thank you, Anya. Let's just hope I live up to your very, very nice introduction. I'm going to be talking about the ocean carbon cycle and how the Benguela system the Benguela Upwelling System feeds into it. So if we look at the global carbon cycle, the reason why it's of such great importance is because the Earth's climate is literally linked to the carbon cycle. That's why if you look at most areas, well, at this point in time, you need to be aware that climate change is a problem. And that's why there's a lot more interest and input that goes into the carbon cycle. And if you looked at one of the problems, which is the carbon dioxide, you actually have higher concentrations in the atmosphere than what we've ever had within like 800 years, so to speak. So the carbon dioxide feeds into global warming, and that cycle is what actually causes the climate change. Now, if we had to take a look at the carbon cycle and actually look at the budget of the sources and the sinks of the carbon, this is the most recent figure I could get. So they basically took the data from 2010 to 2019, and they formulated a budget where you have your arrows having the carbon dioxide that's released, and the arrows pointing down what is actually taken up. So the first thing you notice from this whole graph representation is the fact that, as expected, fossil fuel burning is what has the highest input of carbon dioxide into the atmosphere. Now, if you look at what would be taken up by vegetation due to photosynthesis in terrestrial areas and marine areas, if you combine those, it's nowhere near what you would get from what is actually released by fossil fuels. So we will always have an excess of carbon dioxide. But the next thing you need to notice also is if you look at the bottom, the circles at the bottom essentially give you an idea of the carbon sink. So from that aspect, the largest carbon sink you actually have is in the ocean. And this is in dissolved inorganic carbon. So this gives you an idea that with respect to the global carbon cycle, the oceanic carbon is of great importance. If you compare coastal areas versus offshore areas, the difference in magnitude is actually very high, very big. So if you look at the sink itself, there are two processes that will actually occur in order for you to get the carbon dioxide from the water column and offshore, where it actually sinks to the bottom. And this is through physical and biological processes. And the aspect of it would be to reduce the dissolved organic carbon in the water column and take that off to the offshore. So for the physical processes, you use a solubility pump, which results from ocean circulation. And then the second process that you have is the biological pump, where you actually have the uptake of carbon dioxide and its movement offshore, where it actually goes. And this whole process is done via the food web. So that would include photosynthesizing organisms, as well as the organisms that feed on them, as well as the microbial community that utilizes particulate organic matter. So for the solubility pump, the reason why it actually works is that you have two aspects that basically work together, in the sense that you have high CO2. Okay, cold water is able to take up a lot of carbon dioxide. So that in relation to the ocean thermohaline circulation, you'd end up with a subduction zone with the cold water. So you have the polar latitudes that are moving this water deep into the deep ocean. So that way you'd actually have the movement of carbon so that it ends up in the sink. Now, the problem with this sort of aspect of relating it to a carbon sink is the fact that it takes quite a long period of time. So you're looking at scales of decades to like over 1,000 years. And if you look at the amount that it actually adds to the actual sinking of carbon dioxide, the actual sinking of carbon, it's only about 10 to 35%. So it's not exactly the best. Well, I mean, it happens as it is, but if you wanted to have a process that is more efficient at getting the movement of DIC from the coastal areas right up to the deep ocean offshore, this would necessarily not be it. But 10 to 35 is not bad. Now, the one that is the more efficient option is the biological pump. So this is where you'd actually have your autotrophic organisms that utilize the sunlight, have the DIC in the water, so it's from the dissolved carbon dioxide, and that's taken up and you produce particulate organic matter. And within this area also, you can also have particulate inorganic carbon, and then that would be used for the species that need to form like carbonate shells. And this process has a greater effect on the movement of carbon down to the deep ocean because it amounts to between 65 to 90% of that. So because this is a biological process, it stems to reason that there would be a level of sensitivity because its efficiency is dependent on the factors that actually affect it at the end of the day. So if it gets destabilized, then you would actually end up, instead of taking up the DIC, you would actually release carbon dioxide. So it is of great importance. And with respect to the net sink, so because the biological pump is of great importance, it links to the Benguela from the perspective of that. If you listen to a lot of talks that we've had, they've been talking about primary productivity. This is for if you wanted to know what was actually going on in the water column when the carbon dioxide is being dissolved. So you have your fossil fuels that are burning, that gets dissolved in the water column, and that forms carbonic acid. Now, the carbonic acid, because it's in solution, it can actually form the ions of hydrogen ions and bicarbonate ions. Now, of course, when you see hydrogen ions, you think pH, and that is where you get the decrease in pH, and that is where you get the aspects of ocean acidification. But now these bicarbonate ions can also further ionize into carbonate ions. Now, depending on the level of, depending on the pH concentration in that area, it affects how readily the species are able to utilize the carbonate ions. So they might not necessarily be able to take it up. And of course, we've heard of ocean acidification. So this is why it has like a direct impact with respect to calcareous skeletons. But ocean acidification doesn't only affect the calcareous species. What I like about this graphical representation, even though it's quite old because it's from 2013, it essentially looks at the calcifiers versus the non-calcifiers. And the numbers in brackets under all of those is, it denotes the number of publications that stemmed from getting that result. So they looked at the growth, photosynthesis, and abundance, and got a factor that they related to the mean effect size to see whether or not like you'd actually be negatively or positively affected by ocean acidification. So as expected, the calcifiers don't do very well in ocean acidification, but surprisingly so, the coccolithophores, generally with respect to abundance, it doesn't seem to affect them. Neither does their photosynthesizing capability, and it's only with respect to their growth where they're slightly affected. And for our area, the fact that algae and diatoms seem to do quite well during ocean acidification means that that's an area that should probably be studied in the Benguela. Now, if we look at the Benguela, it is one of the 4 eastern boundary upwelling systems, which we have here in these areas. And this graph representation is looking at chlorophyll concentrations. So even though these areas account for a small surface area with respect to the ocean, they tend to have a high level of primary productivity. So globally, they account for like 2% of the total primary productivity. So because previously we spoke about the fact that the biological pump has such a high, affects the carbon cycle in to such a great extent, these are areas that have to actually be assessed with respect to that. So they're very important in that aspect. So you'd have a very intense biological pump in these areas. And because it's biological, we're dealing with things growing. The seasonal upwelling, which would give nutrients, which would be linked to the growth, would have an effect on the biological pump itself. And the upwelled water tends to be low in pH because it's very cold, so it can take up a lot of carbon dioxide. So the reason why the Benguela is of such significance in this, with this respect, is that of the 4 eastern boundary upwelling cells, the Benguela accounts for 40% of the primary productivity. So the biological pump here is actually very, very strong. And I can't remember who it was, the guy from IOVER, where he was talking about the Benguela currents, where it actually is split along littorets, where you have the northern Benguela and the southern Benguela. So when they looked at air-sea flux data, which was basically, you know, with ships going up and down and them collecting data randomly, they actually realized from their calculations that the northern Benguela is a carbon source, whereas the southern Benguela is a carbon sink. And one of the reasons why this is probable is the fact that you have high remineralization rates in the northern Benguela. But one of the other aspects also is from the fact that we have high phosphate content here and low nitrate. So the nitrate is the limiting factor with respect to primary productivity, as Anya mentioned before. So because these values for the northern Benguela were based on offshore samples, we don't really have an idea of what the variation is in coastal areas, or even just an idea of seasonality and how the upwelling actually affects that source of sink aspect of carbon dioxide. And because it's a biological system, the extent and the influence that this pump would have is also dependent on the species diversity, as well as the composition of the phytoplankton community. So because we're talking about phytoplankton, and if you have species that are able to form blooms, they will proliferate a lot greater. So you have you'll take up more dissolved inorganic oxygen, and you'd have a high amount of particulate organic matter. So these conditions are also linked to what you would see in relation to bloom formation. So you would have, you know, light stratification, all of these factors that would also lead into having a biological pump that's more efficient. So basically taking up DIC quite readily and moving it offshore. Now for this area, because we're in an upwelling system, the strength and the duration of the upwelling, as well as the aerobic decomposition, because that particular organic matter has to be broken down in some way, those processes will also aid in increasing the DIC. So that's the remineralization over there. And as Anya mentioned, we're in a nitrogen deficient area, depending on where the source water comes from, but then that also has a limitation on the amount of dissolved inorganic carbon that can actually be taken up by the phytoplankton. So with this in mind, it took quite a while, but in 2017, there was the Nansen project where they were actually looking at trying to have an ecosystem approach to fisheries. So one of the cruises that they had was looking at pelagic fisheries, and they also were taking samples, you know, it's normal CTD samples. And two of the other factors that they added into that was looking at pH and total alkalinity. So this was the first time we actually got a large data set of carbonate chemistry parameters, in this case being the pH and the total alkalinity. So this was in the 2017 cruise and the 2019 cruise. So what stemmed from this project, even though Corona tried to kill it, but it just moved on to probably next year, is the fact that of the themes that they had, we're able to get funding for a postgraduate student. So Cecilia will be doing her master's. And this is a Norwegian funded project where we're looking at the biological pump, but then also trying to identify and quantify the primary producers in there and to see how much they actually take up with respect to the DIC. And then also look at how the carbonate chemistry affects the calcium carbonate saturation state. So this would be linked to, let's say, your coccolithophores. So those would be, the effects of that would be more offshore than anything else. So the advantage of this is that with the previous cruises, they were focused more on fish with additional sampling. This will cater for more of the actual oceanographic sampling as well. And the other part that was quite good about this is the fact that this project was going along the African coast. So we'll be able to utilize the data collected from this area and relate it to, let's say, off the coast of Morocco. And also, we'll basically be able to compare a lot of things because it's the same ship and the same protocols that are used as you go down the whole coastline. And the other avenue that came up was the fact that the BCC realized that ocean acidification was something that was a global issue, and we didn't really know where we stood with respect to that, even though South Africa is further ahead than everyone else because they do actual regular monitoring. So what they decided to do, because they're part of the BSLME with Angola, Namibia, and South Africa, they essentially decided that they would actually fund an ocean acidification project. So the idea of it is to be able to address the unique problems from each of the different countries, as well as see what we have in common. So for Namibia, we'd also be looking at multiple stresses, which would be the oxygen minimum zones, as Anya spoke about, and also the hydrogen sulfide. And then the industry that is affected by ocean acidification would be the mariculture industry, and that is the industry that is the same for all three countries. So the idea behind it was that we were supposed to start in 2020 and then have a combination of doing sampling at sea so that we can actually get an idea of the seasonal cycle, as well as the variation you get between coastal versus offshore sites, and then also looking at how the carbonate chemistry actually impacts the different organisms in this ecosystem. And then most importantly was the fact that there was enough funding in this project to be able to actually get reference materials that we'll be able to actually use and get some quality assurance in order to make sure that our data is actually what it's supposed to be. And this will also be the first time that we'd actually set up biological experiments and see how that goes. So for South Africa, they essentially are continuing with what they have and then building on top of that. So their data set, they have one in St. Helena where they have mooring systems and they're able to take, temperature, DO, and all these other aspects, and they're able to measure pH, alkalinity, and dissolved inorganic carbon. So that would basically just continue. And then their aspect would be to add the biological OA experiments. And then for Namibia, we would essentially start up the actual monitoring, where we would focus on looking at pH and total alkalinity, together with all the other oceanographic parameters. And this would feed into answering that seasonal coastal versus offshore question. And then for the biological experiments, it would be looking at microalgae and macroalgae microalgae sort of looking at the potential carbon uptake depending on specific species, and also how ocean acidification would affect toxin production, those other aspects. And with that, I am done. Are there any questions?
Okay, thank you, Chibo. Quite interesting. Probably didn't catch you quite well when you mentioned, or when you said the northern Benguela is a carbon source, while the southern Benguela is a carbon sink. I would like you to elaborate a bit on that, and perhaps also bring in the question of carbon sequestration, especially here, perhaps in the Namibian part, which is, I think, the northern Benguela, how we are doing in that regard.
Okay, for the reason why the northern Benguela is a source is because if you look, we have high primary productivity. That gets broken down as it sinks down the water column. So as it's sinking down, we have a large microbial community that decompose at a very high rate. So they essentially break up that particular organic matter and it goes back up into DIC. So that DIC will not necessarily be taken up by the phytoplankton in that water column. So that is what gets released. But again, as I mentioned, this definition of it being a source was related to offshore samples. And offshore is where you have the, what do you call it? The biological pump offshore is not as strong as it is coastal wise. And with respect to carbon sequestration, for Namibia, at the moment, the only project that I know where they're trying to do something similar to that is, I think it's called Blue Kelp, where they're essentially going to grow one species of kelp in a specific area, measure the carbon chemistry, and they'll be able to tell how much carbon they're actually taking up. But then that the biomass that's used has to be utilized somewhere else. Because if it decomposes, you basically just get into the same cycle itself.
Okay, so that's kind of artificial, so to say. But yeah, when you say we are source, it means we perhaps the natural process of carbon sequestration is not so, what do I want to say? It's not so positive, so to say.
I mean, it depends on, okay, because if you look at our system, so we've decided, fine, we say you're a source. Remember, the magnitude of carbon that's released in comparison globally is relatively small. But again, it still doesn't change the fact that we're still releasing carbon dioxide. I would be more comfortable if we had more information on the variability, because it could be much worse than we think, because it's also linked to upwelling, basically. So yeah, I'm a little hesitant to say that it's a waste of time, basically. Yeah. But again, if you look at carbon dioxide, we are not the problem. It's other areas that are emitting fossil fuels. They need to just calm that down also. So maybe they should do something first. And by those people, you know who I mean. Europe, China, the US, you know who I mean. Yeah.
If I can chip in as well. I think Chibo answered quite well there, but she touched also on the issue, Paul, that with the upwelling period, it's very much a seasonal thing also, how strong a source we are or not. You know, it depends on if it's the upwelling or the stratification period for us in the Northern Benguela. And I do know there's a new paper submitted by Claire Siddiqui, and that's part, falls under this traffic project that I alluded to earlier, that I think will give some insights into that, but I don't want to take away about that, but be on the lookout for that paper.
Thank you, Chibo and Anya. Are there any other questions?
I very much like to listen to you, Chibo, but I'm very much also not convinced that the Benguela is a sink or source. It's a continuous recycling of the carbon. The only way you could become a sink is to turn off the nutrients. The nutrient flow produces so much productivity that gets you fixed and degraded again. So that goes in the direction what Paul was asking. But I'm more interested in your experiments. because you're not going to do now experiments to show either that you have a net fixing of CO2 or you have a net release. I wonder what kind of organisms you are using, and are those organisms really relevant for the Benguela population, which are mostly diatoms?
Well, the first phase, I'm going to do a mixture of 3 species of dinoflagellates versus 4 species of diatoms. Now, there's going to be a size factor, and then there's also the fact that the diatoms tend to grow much faster. And also, if you look at the carbon content of diatoms versus dinoflagellates, diatoms have a higher carbon content. So I'm going to do what I can with the cultures that I have. So I'm looking at it as more relating it to specific species, and then also relating it to a combination of communities. And then the second phase of that will be, let's say, adding in the zooplankton component, and then seeing how much of that, like, their grazing rate as well as the fecal matter that they produce. So those 2 aspects will be by themselves, but that doesn't also factor in the microbial breakdown. But that at the moment is a bridge too far for me, so I'm focusing on those 2 aspects first.
And within that program, is someone focusing on the degradation in order to balance at the end, whether it's a sink or source?
You see, that's the thing. If I look at the parameters that we have at the moment that we can measure for the carbon chemistry is pH and total alkalinity. So we can use those parameters to calculate DIC and POC. So we're going to look at those parameters as well as looking at, the growth rates and the change in structure, because some of them also have changes in structure and those aspects. So I'm trying to link as many things as possible, but then I have the limitation of what I have to actually do the analysis with. And actually, one of the things we're also adding now, because we're linked to the Northwestern University, where they tend to do sediment and dust input aspects. So we're going to do a bigger experiment, but then look at dust input and then see how that actually basically affects the whole primary production. But yeah, it's going to be complicated, but yes.
Yes. Yeah, the TOC, because your zinc is the organic carbon. You don't produce any inorganic carbonate.
Yeah, but it's I guess I could filter that off, but then I also need to be cognizant of all the samples that I'm taking.
Sure.
In order to do all those different aspects. But I guess TOC, I mean, that would just be fixing on a filter and then combusting that off.
Yeah.
Could be done.
Well, with regard to your seaweeds, isn't Latoya going in that direction?
I think Latoya is focusing on natural cultures, and I am going to be putting them in aquaria and growing them. So more like vegetatively, not like, actual culture.
Are you doing all phytoplankton, or you mentioned the mariculture industry, are you doing anything with the oysters as we began long ago?
I would like to, but I need to see where that fits in. I was contemplating actually linking that to the seaweed experiment. So basically look at the different species, see which ones are able to actually, affect the carbonate chemistry, and then use that water to feed into the larva. Because Manuel has problems, but because we don't have data on the carbonate chemistry, we can't really pinpoint what it actually is.
Sure.
So I thought that could be one of the ways of fitting it in, basically. Yeah.
Would be useful.
Thank you very much, Chibo, for your talk. Thank you, everyone, for attending today. I think we had a lot of questions, and we look forward to next week when Luke Bridgestock and David Wang will be talking more about micronutrients. Yeah. So until then, we sign off. Thank you very much, everyone.